Yeah, so I'll share my screen. Give me a moment. Can someone unmute and let me know if my screen is visible and if I'm audible? Is my screen visible? Am I audible? You can just type also if my screen is visible and if I'm audible. I'll know, I'll just start. Am I audible? I'll start. So, good evening everyone. Today is the second week of live sessions. Basically, what we do in these live sessions is that we discuss some of the problems from last year assignment questions, and we not only discuss the problems, we also discuss the solution to this problem with the entire concept in mind. Okay, so the purpose is to understand the entire concept so that when we are doing when we are facing similar type of other questions, we can answer from our knowledge. Okay, so that is what these sessions are all about, and we try to keep these sessions as interactive as possible. So if you have any doubt at any point, you can just raise your hand or and unmute your mic to ask me, or you can type it in the chat box. In case if I know the answer now, I'll tell you immediately. If I don't, then you might have to wait till next week. I'll read about it and let you know, but I'll definitely let you know. So my name is Aishi and I'm a PMRF research scholar and from the Department of Chemistry, IIT Bombay. So this is the second session of this course, Pathogen Interaction, with the course instructor Professor Himanshu Kumar from IIT Bhopal. So we'll move on to the first question for today. And just to be clear, all these slides will be uploaded on the NPTEL channel with time, and all the recordings of all these sessions will also be uploaded on YouTube for your viewing. So in any case, if you miss at any point because of weak internet issues or weak network, it's okay, you can just go back to the YouTube videos and check this out. Okay, so going to the first question for today. What is the primary mode of transmission for the HIV? Option A, airborne. Option B, waterborne. Option C, vector-borne. Option D, sexual contact. Option E, foodborne. So I'll wait for a few minutes for you to come up with the question and with the answer, and then I'll move on to the answer with detailed concept about HIV. If you know the answer, you can tell me. Anybody? Anybody? Okay, so it's basically sexual contact. HIV is nothing but human immunodeficiency virus, okay, and it causes AIDS, okay, acquired immunodeficiency syndrome. So basically, it's transmitted when infected body fluids enter the bloodstream or come in contact with mucosal surfaces or damaged tissue of another person. Okay, sexual contact provides the most common and efficient route for this to occur. Okay, and key body fluids that can transmit HIV can also be blood, semen, preseminal fluid, vaginal fluids, rectal fluids, breast milk. So during unprotected intercourse, these fluids can cross mucosal barriers or enter through microscopic tears in the genital or rectal lining, leading to infection, right? And there are some other important but less common transmission routes as well, such as blood transfusion or shared needles, or mother to child transmission during pregnancy, childbirth, or breastfeeding, or occupational exposure like needle stick injuries in healthcare settings. So the learning objective of is to basically know about the basic facts about the impact, trends and definition causes of HIV, to understand the factors that affect the risk of transmission and vulnerability to HIV infection, to discuss strategies to prevent and treat HIV and AIDS, and also care for people with HIV, and we describe the challenges of managing HIV. So these are basically the learning objectives, knowing the basic facts about the impact, trend and definition and causes of HIV. Also, we'll talk about the factors that can affect the risk of transmission and vulnerability to HIV infection. Also discuss strategies to prevent and treat HIV and AIDS and care for people with HIV, and also we'll describe the challenges of managing HIV and AIDS. So as I said, human immunodeficiency virus, it's responsible for causing AIDS. AIDS means acquired immunodeficiency syndrome. Okay, characterized by signs and symptoms of severe immunodeficiency. So there occurs a weight loss of more than 10% of body weight, diarrhea lasting longer than 1 month, fever lasting longer than 1 month. There are other signs as well. Okay, so it was first recognized in 1981 and it's one of the largest epidemics of the 20th century, right? Approximately 33 million people living with HIV by the end of 2007 and 2.7 million people were infected with HIV in 2007. Okay, so will not go. So how does the virus affect? So it basically attacks white blood cells that fight infection in healthy people. It changes the composition of white blood cells and enables HIV to replicate itself. It weakens the immune system. It increases the vulnerability to life-threatening illnesses. Okay, so basically it leads to progressive immune suppression, increases susceptibility to opportunistic infections and cancers, and it's a major public health, social and economic burden worldwide. So as I said, these are the main routes of HIV transmission. Either sexual, that's the main mode of transmission, accounts for 70 to 80% of all HIV infections. It can be parenteral, like blood transfusions. Estimated risk from a single unit of HIV infected whole blood is greater than 90%. Contaminated needle pricks, needle sharing among HIV infected drug users, all of that. It can be vertical, that is perinatal. In utero, like during labor and delivery, and through breastfeeding from mother to child. Now, what are the physical factors that affect transmission and vulnerability? Viral, obviously strain of HIV and viral load. Host, like presence of sexually transmitted infection. Stage of infection, circumcision, hereditary resistance to HIV, all of these. And what are the social and demographic factors in transmission? Social is like views towards women. Um, Basically, demographic is established presence of HIV in the community, mobile and vulnerable populations influencing frequency of partner change, all of that. So, what are the phases of HIV infection? Acute will be HIV replicates quickly, energy requirements increase, asymptomatic because it has no symptoms. Symptomatic can be onset of opportunistic infections, like further increase in nutritional requirements. Late symptomatic heightened viral load, intense weight loss and wasting, and opportunistic infections taking control. So, how is it tested? Maybe it can even turn negative during the first 3 months after infection. That is called the window period. 2 tests are required plus a third to confirm if results conflict. And repeated tests recommend 3 months after initial test. So false positives very possible in children under 18 months old. Okay. So what are the blood tests that for antibodies against HIV? Most common will be enzyme linked immunosorbent assay, ELISA. Gold standard is basically Western blot, which is even more specific than ELISA and used as a confirmatory test. Then there is radio immunobinding assay, which is very expensive. It is used as a confirmatory test when antibodies are very low. Dot blood immunobinding assay, which is very cost effective, rapid screening blood test. We have PCR, which basically polymerase chain reaction, which measures HIV genetic information. So the urine test for antibodies in urine, not as sensitive as blood tests, enzyme immunoassay, EIA, test in blood. Oral fluid test will be test for antibodies in oral fluids, EIA and Western blot, all of these. So what is the treatment of HIV in AIDS? To no cure till now. Treatment can help slow progression to AIDS, reduce OIs and minimize malnutrition. The most effective treatment is antiretroviral therapy using antiretroviral drugs. So, what do you mean by antiretroviral therapy for AIDS, and what are antiretroviral drugs? So, basically, antiretroviral therapy is the standard treatment for HIV infection, including people with AIDS. It involves a long-term, lifelong use of a combination of medicines that control HIV replication in the body. Okay, it does not cure HIV, but it suppresses the viral infection, viral replication. It reduces viral load to very low or undetectable levels. It preserves and restores immune function. It prevents progression from HIV to AIDS, and it reduces HIV transmission. Okay, and what are antiretroviral drugs? These are basically medications that target specific steps in the HIV life cycle, like preventing the virus from multiplying inside the host cells. So HIV replicates through several key steps like entry, reverse transcription, integration, transcription, assembly, maturation. So these which are ARV, basically antiretroviral drugs, they block these steps. Okay, the major classes of antiretroviral drugs can be reverse transcriptase inhibitors, which block conversion of viral RNA into DNA. It can be protease inhibitors that prevent maturation of new viral particles. They can be integrase strand transfer inhibitors, which basically block integration of viral DNA into host genome. They can be entry and fusion inhibitors, prevent HIV from entering host cells. Now, why combination therapy is used? So, basically, 2-3 drugs from different classes are used together. It reduces viral replication more efficiently. It prevents development of drug resistance, and it improves long-term treatment outcome. Okay. So, even in advanced diseases, this is strongly recommended, leads to immune recovery, reduces opportunistic infections and mortality. Okay. So, basically combination of ARVs. We will not go into the demographic too much. This So basically HIV attacks and weakens the immune system, reducing its ability to fight infection. Main transmission routes are sexual, parenteral, vertical. The 4 phases of HIV disease are acute, asymptomatic, symptomatic and full-blown AIDS. HIV is considered recognized mainly by testing antibodies formed to fight the virus. Antiretroviral therapy can prevent OIs, opportunistic infections, and slow progression of AIDS. A comprehensive package of HIV and AIDS interventions should include infection prevention, antiretroviral therapy, care and support, and mitigation of the impacts of HIV on food security, health, education, all of that. Okay, so we already talked about these. So when the immune system becomes weakened by HIV, the illness progresses to AIDS. And some blood test symptoms of certain infections indicate progression of HIV to AIDS. Okay. So you can see it's associated with similar opportunistic infections as well. Basically, our immune system gets so weak that other pathogens can also infect, which would otherwise not, which otherwise your body would have fought off. In case of AIDS, your body becomes so weak, the immune system becomes so weak that your body can no longer fight some easy pathogens as well. Okay, HIV 2 is easily transmitted, it's less pathogenic, duration of HIV 2 infection is shorter, all of these. So as I said, the transmitted by direct contact with infected blood, sexual contact, direct contact with seminal, vaginal and cervical secretions from infected mothers to infants during pregnancy, delivery or breastfeeding. It's not transmitted by coughing, sneezing, insect bites, touching, hugging, all of these. Okay, public wash, handshakes. People have misconception about all these, so we'll not go. So some of these are very demographic results. We will not go into too much detail. So prevention of HIV transmission, basically public health strategies that have been taken to prevent HIV transmission are screening of all blood and blood products, universal precautions should be followed, safer sex practices should be preached, we should identify and treat STIs and other infections, all of these. Now what happens natural course of infections? If you can see in months, at first there will be acute infection. More than half of the HIV infected individuals experience flu-like symptoms during this stage of infection, called the seroconversion illness. Then the immune system helps reduce the viral load for a long period of time, but replication is constant. Then as viral load will increase, disease will progress. So opportunistic infections occur as CD4 plus T cells count drop. We have talked about CD4 plus T cells in our last class, first class. Basically, immune system works to remove the virus from the blood. So as I said, virus can be transmitted during each stage, either from the seroconversion, basically infection with HIV, antibodies develop, asymptomatic, there is no signs of HIV, immune system controls virus production. Symptomatic is physical signs of HIV infection, some immune suppression. And AIDS is like opportunistic infections and stage disease, right? So as I said, HIV will attack the white blood cells called CD4 cells that protect body from illness. Over time, the body's ability to fight common infections is lost, and opportunistic infections which otherwise would not have occurred, or which otherwise the body would have fought and countered, they also occur. Okay, so progression of HIV disease is measured by CD4 plus count, which is a degree of immune suppression. Lower CD4 plus count means decreasing immunity. Then viral load, amount of virus in the blood. Higher viral load means more immune suppression. So severity of illness is determined by the amount of virus in the body, increasing viral load, and the degree of immune suppression, that is decreasing CD4 plus counts. Okay, so higher the viral load, the sooner immune suppression occurs. Okay, so what is the high viral load? The number of copies of HIV in the blood, low CD4 count, the type of white blood cell, increasing clinical symptoms such as opportunistic infection. These are all regions of progression of HIV infection. So the direct infection of organ systems, HIV can directly infect brain, which will cause HIV dementia, or gut, which can cause wasting, and heart, cardiomyopathy as well. So HIV multiplies inside the CD4 plus cell, summarizing and destroying them. As CD4 plus cell count decreases and viral load increases, the immune defenses are weakened. HIV infected people become vulnerable to opportunistic infections. HIV is a chronic viral infection with no known cure, and without antiretroviral therapy, antiretroviral drugs, HIV progresses to symptomatic disease and AIDS. So this is, HIV is a global pandemic, and the number of people living with HIV continues to increase worldwide. Its epidemic is especially severe in resource-constrained settings. HIV is a virus that destroys the immune system, leading to opportunistic infections, and the progression of initial infection with HIV to end-stage AIDS varies from person to person and can take even more than 10 years. The most common method is obviously heterosexual transmission. Women of childbearing age are at particular risk for acquiring HIV through unprotected intercourse, And HIV positive women who are pregnant are at risk of passing the HIV infection to their newborn, right? So risk of HIV transmission from mother to child can be greatly reduced through effective PMTCT programs. Okay, so it's probably infection is probably lifelong, and severe immune deficiency will develop up to half of the HIV infected adults within 8 years after infection. And once severe immune damage develops, the infected person is susceptible to many opportunistic infections and cancers, and these illnesses surrogate or indirect indicators of the immunodeficiency due to HIV, and collectively they constitute the diagnosis of AIDS. And annual progression rates from HIV infection to the development of AIDS is similar in all these countries. So these we have already discussed, transmitted from person to person primarily via blood or other body fluids that may contain some blood, and basically the risk is directly proportional to the amount of blood exchanged. The risk of transmitting HIV infection via blood transfusion from an infected donor is close to 100%, whereas the risk of an HIV infected female transmitting HIV to her partner can be as low as 1 per several thousand episodes of intercourse. So, in the absence of facilitating factors, sexual HIV transmission is several hundred times less infectious compared to most other sexually transmitted agents. Okay. Okay, we will not go into too much detail. This is too much about numbers. We started with this. We are summarizing now. So the question that we started with was, what is the primary mode of transmission for the HIV? The options were airborne, waterborne, foodborne, vector-borne, sexual contact. So the answer will be sexual contact. Any question? Yeah, it's 4. And if you have any question, you can ask me now, or we'll move ahead to the next question. I'll wait for a few minutes and we'll move to the next question for today. Okay, so if nobody has any question, we will move to the next question for today. Which immune cell is responsible? I'll read. Which immune cell is responsible for engulfing and digesting pathogens? A. T cells, B. B cells, C. Macrophages, D. NK cells and natural killer cells, E. Dendritic cells. And if you answer, you have to answer in detail. I'll wait for a few minutes. If anybody wants to try, you can just let me know. Otherwise, I'll move ahead. Anybody? Yes, you're correct. It will be. One second. It will be macrophages. So, one second. So, what do we mean by engulfing and digesting pathogens? Basically, this process is called phagocytosis. So, cells that perform phagocytosis, basically, what they do is that they recognize pathogens, they engulf them by extending their membrane, they digest them using lysosomal enzymes once the phagosome and the lysosome fuse. So, cells specialized for this function are called phagocytes. And why macrophages are the correct answer? Because macrophages are derived from the monocytes in the blood. They reside in tissues like lungs, liver, spleen, brain, etc. They can act as professional phagocytes. Their functions will be engulfing and digesting bacteria, viruses, dead cells, and debris, killing pathogens using lysosomal enzymes, reactive oxygen species, and nitric oxide. They can act as antigen presenting cells or APCs as well by presenting pathogen fragments on MHC class II molecules to T cells, and they can secrete cytokines like interleukin 1, TNF alpha like 2 necrosis factor alpha to regulate inflammation and immune responses. So, because engulfment and digestion are their primary and defining functions, macrophages are the best answer. Okay. So, if we talk, we talked about blood, like the components of the whole blood we talked in the last class. Okay, so if you withdraw blood and place in tube and you centrifuge it, you get plasma, which is 55% of the whole blood. Then we'll get a buffy coat in the middle, which will have your leukocytes and platelets, that is less than 1% of the whole part. And then we'll have erythrocytes, 45% of the whole blood. So, what are the components of blood? Composition will be have plasma 55% and cells. Among the cells, there is WBC, RBC, and platelets. Okay, this is how it works. Plasma will have water, ions, organic molecules such as amino acids, proteins, glucose, lipid, nitrogenous waste, protein, trace elements, and vitamins, also gases such as CO2 and O2. And cellular elements will be red blood cells, white blood cells, and platelets. White blood cells are categorized into so many different types. We talked about this in last class. Granulocytes and granulocytes, right? So, obviously, the function of blood will be carrying O2 and nutrients from the cells, carrying waste from all cells to elimination sites like lungs for CO2, liver for bilirubin, and kidneys for nitrogenous waste. It also carries hormones from endocrine organs to target tissues. So basically, there's a lot of regulatory functions like body temperature, absorbing and distributing heat, pH by virtue of its many buffers, maintains adequate fluid volume in the body, Protective functions will be preventing blood loss by initiating clotting mechanisms in response to blood vessel damage, prevents infection via WBCs and plasma immune proteins. Now, separation of plasma proteins can be done via various technology, basically techniques. If we focus on solubility, there is this method called salting in, salting out. If we focus on molecular size, we have dialysis, gel filtration or size exclusion, chromatography, ultracentrifugation, SDS, gel electrophoresis. In case of molecular charge, we have ion exchange chromatography, high performance liquid chromatography, electrophoresis. And as for specific binding of the protein to a specific substance, that can be affinity adsorption chromatography, precipitation by antibodies. Okay. So like protein solubility, like salting out will basically mean adding divalent salts like ammonium sulfate. Salting in will basically dialysis against a solution with low salt concentration. These are for molecular. So if you see, this is what dialysis looks like. At the start we have this, but due to osmosis, the solvent will come outside. This is what we call bottom dialysis membrane, and this is what we get at equilibrium. Gel filtration chromatography is nothing but passing a solution through various beads. So this is the column bead that we see. So the larger particles will not get inside many of the pores. They will only get inside in the large pores and they will come up faster. The smaller ones might get inside very intricate pores, which the large ones are not able to. And so they will take a long time to come out of the column, get eluted. Okay. SDS also, basically you denature the sample with sodium dodecyl sulfate, and the SDS coated proteins are then, it's an ionic detergent that can bind stoichiometrically to number of peptide bonds and separates on the basis of molecular weight. That is how SDS page happens. Okay, so we talked about chromatography matrices. Now, separation procedures we can also talk about immunoelectrophoresis, which is basically a double immunodiffusion technique. Sample is placed in a well on a glass slide coated with agarose cellulose acetate and electrophorese to separate the proteins according to their charge. Okay. A trough is cut in the middle of the agarose parallel to the axis of the electrophoresed proteins, into which it is placed monospecific antibodies against IgA, IgM, IgG or kappa and lambda light chains. The slide is incubated for 18 to 24 hours to allow the antibodies to diffuse from the trough into the agarose, forming precipitin arcs with their respective antigens. And the relative size of the precipitin arcs is proportional to the quantity of immunoglobulin or light chains. ELISA, this is the principle of ELISA. Basically, wells of the ELISA plate will be coated with the antigen. The primary antibody will come and react with the antigen, and reagent added that is converted to colored product by enzyme. In case of competitive ELISA, primary antibody will react with bound antigen and varying amounts of free antigen. And primary antibody bound to antigen detected using secondary antibody that is coupled to an enzyme called peroxidase. So basically, it's binding the antigen to the surface of the wells of an ELISA plate, blocking any non-specific antibody binding sites with a generic protein like BSA, ovalbumin, gamma globulin, skin mill, adding primary antibody solution to each well to allow specific antigen antibody recognition binding to occur and removing unbound primary antibody. So adding a secondary antibody like goat, anti-mouse, anti-IgG that is covalently linked to a protein such as horseradish peroxidase or alkaline phosphatase. This antibody will bind to the primary antigen that remain bound to antigen after washing the wells and remove unbound secondary antibody. You can determine the amount of secondary antibody bound to the wells by adding a chemical reagent that is converted by the peroxidase or the phosphatase to a colored product, basically chemiluminescence, whose colorimetric assay, whose absorbance is measured using a ELISA plate reader. So why so much about, I'll not go into much detail about the separations of function of plasma. Let me go to the not this 1 second. I want to discuss that. Wait a second. Anyway, so yeah. So the question that we started with was macrophage, and basically they are the immune cells specialized for engulfing and digesting pathogens, and they play a key role in both innate and adaptive activation of the adaptive immunity. Okay, so that is what we call macrophages. Basically they efficiently perform all these steps and are called professional phagocytes. So they can be named differently in different tissues, like alveolar macrophages in lungs, Kupffer cells in liver. They are derived from monocytes circulating in the blood. They can be microglia in the brain, splenic macrophages in spleen, and their key functions, as I said, is basically engulfing pathogens like bacteria, viruses, fungi, parasites, or dead and dying host cells. They digest the pathogens using lysosomal enzymes. They produce reactive oxygen species and nitric oxide for microbial killing. They do antigen presentation. They basically present processed antigens on MHC class 2 molecules. They can activate CD4 plus helper T cells. They help in they do cytokine secretion like IL-1, IL-6, TNF alpha, inflammation and immune activation, and they can even bridge innate and adaptive immunity. So because engulfment and digestion are their primary defining roles, macrophages are the best answer for this question. Right? So if you have any doubts, you can ask me now, or else I'll move on to the next question and wait for a few minutes, then I'll move on to the next question. Okay, so moving on to the next question for today. What is the function of vaccines in the immune system? Option A, cure ongoing infections. Option B, protection from infection for which vaccine is given. Option C, suppress the immune response. Option D, increase blood clotting. And option E, improve digestion. So answer in detail. Whoever is choosing to answer, please answer in detail, not only the option. I'll wait for a few minutes and then we'll move on to discussing this question. You're saying option B, right? Yes, ma'am. Okay, anybody else has any other answer or this answer? Okay, so basically what is a vaccine? A vaccine is a biological preparation that contains weakened or attenuated pathogens. They can also contain inactivated or killed pathogens and also sometimes purified components of pathogens like subunit vaccines, and also sometimes genetic material like mRNA or viral vectors. And they are designed to safely expose the immune system to an antigen without causing disease. Okay, so the right answer will be protection from infection for which the vaccine is given. So the main function of vaccines is to protect against specific infections by inducing immune memory. So vaccines activate the immune system, stimulate B cells to produce specific antibodies, activate T helper cells and sometimes cytotoxic T cells as well, generate memory B cells and memory T cells, and enable a faster and stronger immune response upon future exposure to the real pathogen. So as a result, the pathogen is neutralized quickly before it can even cause a disease. That's why the option B here is correct. Protection from infection or from severe disease caused by that infection is the intended and proven function of vaccine. So this protection can be sterilizing immunity, like prevents infection completely, or protective immunity, like prevents severe disease and complications. The other options are not right because it does not cure ongoing infection, because vaccines are preventative, not therapeutic. They do not cure an active infection. The treatment requires drugs like antibiotics, antivirals. It does not suppress the immune response. Vaccines stimulate, not suppress immunity. Immune suppression is achieved by immunosuppressive drugs, not vaccines. It does not increase blood clotting. Blood clotting is a part of the hemostatic system. So vaccines do not regulate coagulation. It does not improve digestion because digestion is a functional of the gastrointestinal system. Vaccines have no role in digestion. So the clinical relevance of vaccination is one of the most effective public health interventions. Basically, it prevents infectious diseases, epidemics and pandemics, and protects both individuals and communities. Herd immunity, right? So as I said, we talked a little bit about active and passive immunization in the last class. So passive immunization is basically methods of acquisition include natural maternal antibodies, antitoxins and immunoglobulins. Basically protection transferred from another person or animal. As active immunization is methods of acquisition include natural infection, vaccines, many types and toxoids. It's relatively permanent. So passive immunity can be natural maternal antibody, immune globulin, antitoxin, etc. Active immunity can be natural infection or vaccines or attenuated organisms which come under vaccines, any inactivated organisms, purified microbial macromolecules, cloned microbial antigens, multivalent complexes, toxoid, etc. So So passive immunization can occur naturally by transfer of maternal antibodies across placenta to fetus. Like injection with preformed antibodies, human or animal antibodies can be used. Injection of animal antibodies prevalent before vaccines. So effects, but the problem here is that effects are only temporary because they are basically adding the antibodies. So whatever has happened, that will be countered, but it does not create any kind of memory inside your body to recognize the pathogen again next time it infects. So what are the conditions warranting passive immunization? Deficiency in synthesis of antibody as a result of congenital or acquired B cell defects. Okay. Susceptible person is exposed to a disease that will cause immediate complications. Okay, and disease is already present. In these kinds of cases, do doctors or people prefer passive immunizations? Otherwise, people always prefer active immunization. So, what are the common agents for passive immunization? So, you can see for measles, we have pooled human immune gamma globulin. For snake bite, we have horse antivenin. All of these. So the transfer of antibodies will not trigger the immune system, right? There is no presence of memory cells. So there is no memory created and kept in the host cells. So there are risks included as well because recognition of the immunoglobulin epitope by self-immunoglobulin paratopes. Some individuals can produce IgE molecules specific for passive antibody leading to mast cell degranulation. Some individuals produce IgG or IgM molecules specific for passive antibody leading to hypersensitive reactions. So, in case of active immunization, it's basically natural infection with microorganism or artificial acquisition like vaccine. So, both stimulate the proliferation of T and B cells, resulting in the formation of effector and memory cells. Right? So, the formation of memory cells is the basis for the relatively permanent effects of vaccinations. Okay. So, what are the principles underlying vaccination? So, if we think about the concept of immunity, there is basically self versus non-self. Antigen specificity indicated by presence of effector cells, protection from infectious diseases using these above methods. So, these are some of the vaccinations recommended childhood immunization schedule. If you see from the US 10 to December, if you see these are some of the poliovirus, rotavirus, measles, varicella, all of these. Now, what is the effectiveness of vaccinations? A small percentage of recipients will respond poorly. That is role of genetic determinants. Herd immunity, what do we mean by? So, what do we basically mean by herd immunity? So, majority of the population is immune, so chance of susceptible individual contacting infected individual is low. Okay, so basically herd immunity, also called community immunity, refers to the indirect protection from an infectious disease that occurs when a large proportion of a population becomes immune to that disease, either through vaccination or previous infections. So, because many people are immune, the pathogen cannot spread easily, which also protects individuals who are not immune. Right? So, how this herd immunity works basically? So, in a fully susceptible population, an infected person can easily transmit the pathogen to others. Now, when a significant fraction of people are immune, they do not get infected, they do not transmit the pathogen. This breaks chains of transmission. As a result, even unvaccinated or vulnerable individuals like infants, elderly, immunocompromised are protected. Okay. Basically, what are the key components of herd immunity? Basically, population level immunity. Immunity must be present in a large fraction of the community. Individual community alone is not enough. Then threshold concept. Herd immunity requires crossing a minimum immunity threshold, and this threshold depends on how contagious the disease is. Then role of vaccines in herd immunity. Vaccines safely increase immunity in the populations, reduces disease spread without causing illness, essential for protecting newborns, people who cannot be vaccinated, and immunocompromised individuals. But what are the limitations of herd immunity? Does not work well for diseases with no effective vaccine, infections with rapid mutation, diseases transmitted through non-community routes, like tetanus. Okay, as I said, there are several factors affecting herd immunity. Environmental factors will be crowded conditions, seasonal variations, strength of individual's immune system, infectiousness of disease, like greater the risk of infection, the higher percentage of people need vaccines to attain herd immunity. And when enough people are vaccinated, chance of germ infecting the non-immunized population is small. So can lead to disappearance of diseases like smallpox, vaccination no longer necessary. Now, what are the common misconceptions that activation of the immune system results in protective immunity? Multiple factors affect decisions when making vaccines. Number one is activation of specific branch of immune system. Number two, development of immunological memory. So, what is the role of memory cells? So, it depends on incubation period of pathogen. So, memory cells will need short incubation periods like influenza, symptoms already underway by the time memory cells are activated, and repeated immunizations with neutralizing antibodies. Long incubation periods will be poliovirus, enough time to allow memory B cells to respond. So, this is a graph showing immunological memory versus serum antibody levels. So, what are the types of vaccines? Can be whole organism like attenuated viral or bacterial, can be inactivated viral or bacterial, can be purified macromolecules like polysaccharides, toxoid, recombinant antigen, recombinant vector, or DNA, or synthetic peptide, or multivalent subunit. So, many common vaccines used consist of inactivated or attenuated bacterial cells or viral particles. Basically, it includes attenuated and inactivated vaccines. So, you can see some of the classification of common vaccines for humans. You can see over here, for example, for anthrax, cholera, pertussis, plague, we have inactivated. For tuberculosis, we have live attenuated BCG, Bacillus calmette-guerin. For typhoid, we have live attenuated. For viral particles like hepatitis A, inactivated. Influenza, inactivated. Measles, mumps, live attenuated. All of these. So, attenuation is equal to reduce in force, value, amount, or degree when we can achieve by both under normal cultural conditions. Bacillus calmette-guerin act as a double-edged sword as they have distinct advantages and disadvantages. So, advantage system from their capacity for transient growth, prolonged immune system exposure, single immunization, replication within the host cells. Now, what is the exception to the rule? So, Sabin polio vaccine consists of 3 attenuated strains of poliovirus. So, colonization of intestine results in immunity to all these 3 strains and production of secretory IgA and induction of IgM and IgG. So, the result is the need for boosters. Individual strains interfere with one another. So, the first immunization will be one strain predominates in growth. Second immunization will be immunity generated by previous immunization limits growth of previously predominant strain. Third immunization will be same principle as second immunization. Now, what are the disadvantages of attenuated bacterial or viral vaccines? Major disadvantage is possible reversion, that is, rate of reversion of Sabin polio vaccine is 1 case in a 4 million doses. presence of other viruses as contaminants, and unforeseen post-vaccine complications. So, what is the future of attenuation? Genetic engineering techniques provide new methods of attenuation, like herpes virus vaccine for pigs, possible elimination of reversion. So, what are the methods of inactivation? Will include heat or chemical agents, and resultant loss of replication ability, and difficulty inactivate due to the potential for denaturation of epitopes, dependence on higher order levels of protein structure. So this is a comparison of attenuated live and inactivated killed vaccines. Okay, so for the production, if you see, for the attenuated vaccines, we need selection of non-virulent organisms. Virulent pathogens is grown under adverse culture conditions or prolonged passage of a virulent human pathogen through different hosts. Genetically requires only a single booster, less stable, whereas the inactivated vector vaccine, is virulent pathogen is inactivated by chemicals or in irradiation with gamma rays. Salination is basically normally require one dosage to induce relatively permanent immunity, primarily cell mediated in nature. Despite reliance on cell mediated immunity, increased IgA response, whereas inactivation requires multiple boosters, emphasis on activating humoral immunity. However, something very important is missing, which is adjuvants. So there are two terms that you need to familiarize yourself with: adjuvants and haptens. What are these? What are their functions in detail? So adjuvants are substances that are added to a vaccine to enhance or boost the immune response to the antigen. Okay. Now, why are adjuvants needed? Many vaccine antigens, especially purified proteins or subunits, are poorly immunogenic in their form, rapidly cleared from the body. Adjuvants make the immune system notice and respond more strongly to these antigens. So, what are the functions of adjuvants? Basically, enhance antigen presentation, increase uptake of antigen by antigen presenting cells, dendritic cells, and macrophages. They provide better presentation on MHC molecules. Then activate innate immunity, stimulate pattern recognition receptors (PRRs), gastrin-like receptor (TLR), lead to secretion of cytokines and chemokines, provide the danger signal required for adaptive immunity. Prolonged antigen availability. Some adjuvants create a depot effect. Antigens is released slowly over time, leading to prolonged immune stimulation. Now, if you want to improve quality of immune response, you can enhance the antibody titers, T cell responses, memory cell formation. So, immunosuppressing complexes are also there. Multivariate presence of antigen, presentation of antigen and adjuvant, enhanced cell-mediated immune response, delayed type 2 sensitivity, cytotoxic T lymphocyte response, increased antigen expression associated with MHC2. Now, what is the hapten? We have been talking about adjuvant. Hapten is basically a small molecule that can bind to antibodies but cannot by itself induce an immune response. A hapten is a low molecular weight and hence it is antigenic. Substance that is antigenic but not immunogenic enough unless attached to a carrier protein. So why haptens are not immunogenic alone? Too small to be processed by APCs, activate T helper cells. How haptens become immunogenic? When a hapten binds to a large central protein, the complex becomes immunogenic. Carrier protein provides T cell epitopes Hapten provides basic specificity together full immune response. Functions and importance of haptens: explain drug allergies. Many drugs act as haptens. Drug binds to host proteins. All allergy, all of that. Used in experimental immunology as well to study antibodies, specifically antigen recognition classic examples like FIT-SEAM. Then we do vaccine design, which is basically conjugated vaccines. Example, polysaccharide protein vaccines. They improve immune response in infants. So basically, vaccine components, they provide immunity, antigen adjuvants. The key vaccines are safe and long-lasting, preservative stabilizers used during the production of vaccines, cell culture materials, inactivating ingredients, antibiotics. The antigens are very small amounts of weak or dead pathogens that can cause diseases. They help the immune system learn how to fight off the infections faster and more efficiently. Influenza virus is an example of antigen adjuvants, which are in some vaccine and substances that help the immune system would respond more strongly to a vaccine. So, which are in some vaccines or substances that help the immune system respond more strongly to a vaccine. This increases the immunity and against the disease. Aluminium is an example of an adjuvant. Aluminum is the most abundant metal found in plants, soil, water, and air. So basically, preservatives may be defined as compounds that kill and prevent the growth of microorganisms, particularly bacteria and fungi. They are used in vaccines to prevent microbial growth in the event that the vaccine is accidentally contaminated, as might occur with repeated function of multi-dose vials with the needle. In some cases, preservatives are added during the manufacturing process to prevent microbial growth. However, improvements in manufacturing technology have markedly decreased the needs to add any preservatives during the manufacturing process. So, what are preservatives? Basically, it's a mercury coating containing organic compound, an organomercurial. Since the 1930s, it has been widely used as a preservative in a number of biological and drug products, including many vaccines, to help prevent potentially life-threatening contamination with harmful microbes. The documented antimicrobial properties of uh, thimerosal, for example, contribute to the safe use of vaccines in multi-dose vials and seasonal and pandemic influenza in multi-dose vials that help facilitate immunization. So, vaccine containing 0.01% thimerosal act as a preservative. For comparison, this is roughly the same amount of elemental mercury contained in a 3-ounce can of tuna fish, basically. So, the thimerosal is in concentrations of 0.001% to 0.01% has been shown to be effective in clearing a broad spectrum of pathogens. So, while the use of mercury containing preservatives has declined in recent years due to the developing of new products formulated into single dose presentations that do not require preservatives, thimerosal has been used in some immune globulin complexes, antivalents, and skin test antigens, and ophthalmic and nasal products, in addition to some vaccines. Now, phenol is also used as a preservative in the FDA-approved available vaccines. Pneumonia 23 for preventing of pneumococcal disease caused by the 23 serotypes contained in the vaccine, and typhine 6 for prevention of typhoid fever, and ACAM 1000 for prevention of smallpox. Each of these vaccines contain 0.25% phenol. Now, these vaccines are not recommended for routine use by the Centers for Disease Control and Preventions and Advisory Committee on Immunization. So, phenol is used in a variety of consumer products, including mouthwashes, throat lounges, and throat sprays. So, like sugar or gelatin that have the active ingredients in vaccines continue to work when the vaccine is made, stored, and more. Sometimes trace amounts of some ingredients that are needed to produce the vaccine remain in the final product. Examples of these non-harmful ingredients include like cell culture material or inactivating germ killing ingredients like formaldehyde to weaken or kill viruses, bacteria, or toxins in the vaccine. Antibiotics like pneumycin to help keep outside germs and bacteria from growing in the vaccine. A heterologous vaccine, also known as a Jennerian vaccine, is a type of live vaccines where one pathogen is introduced in order to provide protection against a different one. The vaccines are pathogens of other animals and either do not cause disease or cause mild disease in the organism being treated. Examples will be generous administration of cowpox vaccinia to protect them against smallpox, variola, BCG vaccine made from Mycobacterium bovis to protect against human tuberculosis. Now, homologous vaccines are those which are produced from one and used against the same. So, the vaccine can be classified as routine vaccine. They are well tolerated, provide adequate protection against dangerous and widespread infection. Example: diphtheria, polio, measles, mumps, and most people think of these as childhood vaccines that you get before starting school. But some vaccines are routinely recommended for adults and some are recommended every year, like a flu vaccine, or every 10 years, like a tetanus booster. So, indicated vaccines are performed only under certain conditions. So, these vaccines belong to this group. So, most vaccines in current use work by inducing humoral immunity. The consideration will be safety, immunogenicity, type of protection, time, quickness, longevity, route of administration, magnitude, and antigenic variation. Target population, obviously. Okay. Storage and cost. Now, what are the benefits of vaccine? Protection or immunity, reduce exposure, disease prevention like individual, community, or herd immunity. Cost. Now, what is a genotype recipient? A major factor that determines immune responsiveness. So, some substances are immunogenic in one species but not in another. Similarly, some substances are immunogenic in one individual but not in others. So, MHC gene products which function in processing and presenting antigen influence response towards antigen. Genes that codes B cell and T cell receptor also influences immunogenicity. Also, the genes coding proteins for various regulatory mechanisms influences it. So, an insufficient dosage of immunogen will not elicit an immune response. Either it fails to elicit immune response or lead to a state of tolerance. Conversely, an excessively high dose will also lead to unresponsiveness or tolerance. Okay, 1 second. Why is it not moving? So, single dosage obviously is not efficient to develop immune response. However, booster dose over a period of time enhances immunogenicity. Then administration route strongly influences the immune response. The subcutaneous route is better than the intravenous or intragastric routes. Then adjuvants, as I said, are designed to improve poorly immunogenic vaccines. One way to increase the immunogenicity of vaccine is introduce additional material specifically to provoke a stronger immune response. Then there are substances that when mixed and injected with antigen enhances the immunogenicity of the antigen. Like a group of salt adjuvants in most common use aluminum salts. Now I already discussed adjuvants, basically like aluminum potassium sulfate. Alum is an adjuvant that enhances the immunogenicity by increasing persistence of antigen, like releasing antigen slowly from injection site and enhancing phagocytosis of antigen, TLR interaction, and cytokine induction. So water and oil or oil in water emulsion like fluorines and adjuvant, as well as natural and synthetic toxins derived from bacteria like cholera toxin, CT and lymphotoxin LT, slow release of antigen. Then we have fronds and adjuvant. This is a solution of antigen emulsified in mineral oil and used as an immunopotential booster. Then we have the complete form fronds complete adjuvant. Then we have it's composed of inactivated and dried mycobacteria, usually M. tuberculosis, whereas B will be incomplete form, like lacks the mycobacterial components, hence just the water in oil emulsion. The use of adjuvants, however, is often hampered by undesirable side effects such as fever and inflammation. Like adjuvants enhance immunogenicity in various ways. For example, prolonged persistence of antigen, increase the immunogenicity of weak antigens. Co-stimulatory signals are prolonged and enhance the speed and duration of the immune response. Then we have local inflammatory response, which is increased and stimulates and modulates humoral responses, stimulates cell-mediated immunity, and improve the induction of mucosal immunity. Then we have non-specific proliferation of lymphocytes, which is increased. Now, other issues associated with immunization is antigenic variation. Influenza virus, for example, can rapidly alter its antigenic structure by mutation so that it is no longer recognized by antibodies made against the original virus. Influenza can therefore give rise to repeated infection with variants of the same organism. Then we have antigenic competition. Two antigens given at the same site can sometimes each interfere with the immune response to the other. In general, therefore, different immunizations are given at different times and on different sites. But remember that DPT, that is diphtheria, pertussis, tetanus, is a notable exception to this rule, and other routine multiple vaccination protocols exist, such as MMR. I think measles, mumps, rubella, something like that. Then we have maternal immunoglobulin, basically the presence of specific antibodies at the time of vaccination may interfere with its success. So, the measles vaccine, for example, should not be given before 15 months of age, since the presence of maternal IgG antibodies may prevent active immunization. Now, what is plasma therapy? Plasma is basically the largest part of blood. It makes up more than half of about 55% of its overall content. So, when separated from the rest of the blood, plasma is a light yellow liquid. So, the main role of plasma is to take nutrients, hormones, and proteins to the parts of the body that need it. Cells also put their waste products into the plasma. The plasma then helps remove this waste from the body. So, blood plasma also carries all parts of the blood through your circulatory system. Now, along with water, salt, and enzymes, plasma also contains important components. These include antibodies, clotting factors, and the proteins albumin and fibrinogen. Proteins and antibodies in plasma are also used in therapies for rare chronic conditions. These include autoimmune disorders and hemophilia. Now, several countries are seriously looking at plasma therapy as the potential treatment for COVID-19, the disease caused by the novel coronavirus. Plasma therapy uses blood donated by recovered patients to introduce antibodies in those under age. So, as I said, vaccination is a method of giving antigen to stimulate the immune response through active immunization, right? And basically, it's a immunobiological substance designed to produce specific protection against a given disease. So, a vaccine is antigenic but not pathogenic. So, as I said, there can be live vaccines, attenuated vaccines, inactivated or killed vaccines, toxoids, polysaccharides and polypeptide, cellular fraction vaccines, surface antigen recombinant vaccines. So, live vaccines are made from live infections agents without any amendment. The only live vaccine is variola smallpox vaccine made of live Vaccinia cowpox virus. Not variola virus, which is not pathogenic but antigenic, giving cross immunity for variola. Now, live attenuated or avirulent vaccines, virulent pathogenic organisms are treated to become attenuated and avirulent but antigenic, and they have lost their capacity to induce full-blown disease but retain their immunogenicity. So, live attenuated vaccines should not be administered to persons with suppressed immune response due to leukemia and lymphoma, other malignancies, receiving a corticosteroids and anti-metabolic agents, radiation, pregnancy. Right? Then we have inactivated or killed vaccines. Organisms are killed or inactivated by heat or chemicals but remain antigenic. They're usually safe but less effective than live attenuated vaccines. The only absolute contraindication to their administration is a severe local or general reaction to a previous dose. Now, what are toxoids? They are basically prepared by detoxifying the exotoxins of some bacteria, rendering them antigenic but not pathogenic. So, like adjuvant, like we already discussed in this session, like alum precipitation, is used to increase the potency of a vaccine. Now, the antibodies produced in the body as a consequence of toxoid administration neutralize the toxic moiety produced during infection rather than act upon the organism itself. So, in general, toxoids are highly efficacious and safe immunizing agents. Now, what are polysaccharide and polypeptide cellular fraction vaccines? They are basically prepared from extracted cellular fractions. Example, meningococcal vaccine from the polysaccharide antigen of the cell wall. The pneumococcal vaccine from the polysaccharide contained in the capsule of the organism, and the hepatitis B polypeptide vaccine. Their efficacy and safety appear to be very high. Then we have surface antigen recombinant vaccine. It is prepared by cloning HBsAg gene in yeast cells. Where it is expressed, then HBsAg produced is then used for vaccine preparations. Their efficacy and safety also appear to be very high. Now, these are the types of vaccines, as I said, and some of the examples. For live vaccines, smallpox, variola. Live attenuated will be BCG, Bacillus calmette-guerin. You can read more about it. Typhoid, oral plague, yellow fever. Then killed inactivated vaccines like typhoid, cholera, pertussis, plague. Then toxoids like diphtheria, tetanus. Then cellular fraction vaccines like meningococcal, polysaccharide vaccine, all of that. Recombinant vaccines like hepatitis B vaccine. Now, what are the routes of administration? Deep subcutaneous or intramuscular route, most vaccines. Oral route can be oral BCG vaccine, intradermal route BCG vaccine, scarification, smallpox vaccine, intranasal route, live attenuated influenza vaccine. Now, scheme of immunization, primary vaccination is one dose vaccine like BCG, variola, measles, mumps, rubella, yellow fever. Then multiple dose vaccines, polio, DPT, hepatitis B. Then booster vaccination to maintain immunity level after it declines after some time has elapsed. Now, what are the periods of maintained immunity due to vaccines? Short period cholera vaccine, 2 years TAB vaccine, 3 to 5 years DPT vaccine, 5 or more years BCG vaccine, 10 years will be yellow fever vaccine, solid immunity like measles, mumps and rubella vaccines. Now, about the levels of effectiveness, absolutely protective 100% is yellow fever vaccine. Almost absolutely protective 99% variola, measles, mumps, rubella vaccines and diphtheria and tetanus toxoids. Highly protective polio, BCG, hepatitis B and pertussis vaccines, and moderately protective TAB, cholera vaccine and influenza killed vaccine. Now, what is a cold chain? A cold chain is a system of storage and transport of vaccines at the low temperature from the manufacturer to the actual vaccination site. And the cold chain system is necessary because vaccine failure may occur due to failure to store and transport under strict temperature controls. Right? So there are some cold chain equipments that this is also need to be studied, like walking cold rooms. They are located at regional level, meant to store vaccines up to 3 months and serve districts. Deep freezers and ice-lined refrigerators, then small deep freezers and all of these. Cold boxes which are supplied to all peripheral centers. These are used mainly for transportation of the vaccines. And vaccine carriers is used to carry small quantities of vaccines for the out-of-reach sessions. 4 fully frozen ice packs used for lining the sides. Ice packs contain water and no salt should be added to it. Among the vaccines, polio is the most sensitive to heat, requiring storage at minus 20 degrees Celsius. Vaccines which must be stored in the freezer compartment are polio and measles, and vaccines which must be stored in the cold part but never allowed to freeze are typhoid, DPT, tetanus, toxoid, DT, BCG and diabetes. Now, what are the hazards of immunization? So, no immune response is entirely free from the risk of adverse reactions, right? Or remote sequelae. But the adverse reactions that may occur may be grouped under the following head, like reactions inherent to inoculation, reactions due to faulty techniques, reactions due to hypersensitivity, neurological involvement, provocative reactions, and others. Now, what are the reactions inherent to inoculation? These may be local general reactions. The local reactions may be pain, swelling, redness, tenderness, and development of a small nodule or sterile abscess at the site of injection. The general reactions may be fever, malaise, headache, and other constitutional symptoms. Most killed bacterial vaccines like typhoid cause some local and general reactions. Diphtheria and tetanus, toxoids, and live polio vaccine cause little reaction. Now, what are the reactions due to faulty techniques? Faulty techniques may relate to faulty production of vaccine, like inadequate inactivation of the microbe, inadequate detoxification, too much vaccine given in one dose, improper immunization site or route, vaccine reconstituted with incorrect diluents, wrong amount of diluent used, drug substituted for vaccine or diluent, vaccine prepared incorrectly for use, vaccine or diluent contaminated, vaccine stored incorrectly, contraindications ignored, reconstituted vaccine of one session of immunization used again at the subsequent session. So improperly sterilized syringes and needles carry the hazard of hepatitis B virus and staphylo and streptococcal infection. Now there can be reactions due to hypersensitivity as well. So administration of anti-sera may occasionally give rise to an anaphylactic shock and serum sickness. So many viral vaccines contain traces of various antibiotics used in their preparation, and some individuals may be sensitive to the antibiotic which it contains. Anaphylactic shock is a rare but dangerous complication of injection of antiserum. Okay, there is bronchospasm, dyspnea, pallor, hypotension, and collapse. And the symptoms may appear within a few minutes of injection or may be delayed up to 2 hours. Some viral vaccines prepared from embryonated eggs like influenza may also bring about generalized anaphylactic reactions. And serum sickness is characterized by symptoms such as fever, rash, edema, and joint pains occurring 7 to 12 days of injection of antiserum. There can be neurological involvement as well, like neuritic manifestations may be seen after the administration of serum or vaccine, and the well-known examples are the post-vaccinal encephalitis and encephalopathy following administration of anti-rabies and smallpox vaccines. Guillain-Barré syndrome in association with the swine influenza vaccine is another example. Provocative reactions occasionally following immunization. There may occur a disease totally unconnected with the immunizing agent, like provocative polio after DPT or DT administration against diphtheria, and the mechanism seems to be that the individual is harboring the infectious agent, and the administration of the vaccine shortens the incubation period and produces the disease or what may have been otherwise only a latent infection is converted into a clinical attack. Now, others may be comprised damage to the fetus, like rubella vaccination, displacement in the age distribution of a disease, like a potential problem in mass vaccination against measles, rubella, and mumps. Now, what are the precautions that need to be taken? Before administration of the antiserum or antitoxin, it is necessary to test for a sensitivity reaction. Like this can be done in two ways. Like we can instill a drop of the preparation onto the conjunctival sac, and a sensitized person will develop pricking of the conjunctiva. A more reliable way of testing is by intradermal injection of 0.2 ml of antiserum diluted 1:10 with saline. A sensitized patient will develop a wheal and flare within 10 minutes inside of injection. It should be borne in mind that these tests are not without faults, right? So adrenaline, like 1,000 solution, should be kept ready when giving foreign serum. In the event of anaphylaxis for an adult, 0.5 ml of adrenaline solution should be injected intramuscularly immediately, followed by 0.5 ml every 20 minutes if the systolic blood pressure is below 100 mm of mercury. So why is adrenaline always kept ready when giving any kind of foreign serum to a person? This question might come up in your mind. So because of the risk of a certain life-threatening allergic reaction called, as I said, anaphylaxis. So as I said, foreign serum, we mean anti-sera, which is anti-snake venom, anti-rabies serum, etc. or immunoglobulins or some vaccines or biologically derived products. So these vaccines, these substances contain foreign proteins that the recipient's immune system may recognize as non-self antigens. So anaphylaxis will be a type 1 hypersensitivity reaction, right? Immediate, like seconds to minutes, IgE mediated, potentially fatal. Mechanism behind it: on prior exposure, the person may have developed IgG antibodies against the foreign serum proteins. These IgG antibodies bind to mast cells and basophils. Now, on re-exposure, antigen cross-links IgE on the mast cells, leads to massive degranulation Like release mediators will be histamine, leukotrienes, prostaglandins, cytokines. Now the effects of anaphylaxis on the body, these mediators cause severe vasodilation, sudden drop in blood pressure like shock, increased vascular permeability, swelling, angioedema, bronchoconstriction, difficulty breathing, laryngeal edema, airway obstruction, urticaria, itching, flushing. So death can occur rapidly due to airway obstruction or circulatory collapse. Why adrenaline or epinephrine is the drug of choice? Because it's kept ready, because it counteracts all the major life-threatening effects of anaphylaxis. Okay, it causes vasoconstriction, raises blood pressure, reduces edema and capillary leakage. It can increase heart rate and cardiac output, and prevents cardiovascular collapse. It can cause bronchodilation, relieve airway obstruction, inhibit further mast cell degranulation. So no other drug can act this fast and this comprehensively. Okay. Then an injection of antihistamine drug should also be given by the intramuscular route to minimize the after effects such as urticaria or edema, as I said. The patient should be observed for 30 minutes after any kind of serum injection. So the risk of adverse reactions can be reduced by proper sterilization of syringes and needles, by proper selection of the subject and the product, and if due care is exercised in carrying out the procedure. Like measles and the BCG vaccine should be reconstituted only with the diluent supplied by the manufacturer. Reconstituted vaccine should be discarded at the end of each immunization session and never retained for use in subsequent sessions. In the refrigerator of the immunization center, no other drug and substances should be stored beside vaccines. Training of immunization workers and their close supervision to ensure that proper procedures are being followed are essential to prevent complications and even deaths following immunization, right? So vaccination coverage is the percent of at-risk or susceptible individuals or population who have been fully immunized against particular diseases by vaccines or toxoids to be significantly effective in prevention of disease on mass or community level. At least a satisfactory proportion of the at-risk population must be immunized. Now, what are the ways of achieving satisfactory immunization coverage? Efficient immunization service, urban and rural areas, in towns, cities, and even in villages. Health awareness, right, and cooperation of the public. Periodic mass immunization campaigns to cover those who missed regular immunizations. Outreach programs in rural and nomad areas, and home visits. Now, what is the application of active immunization? Infants and children expanded immunization program. Active immunization for adult females, vaccination for special occupations, vaccinations for lifestyles, special lifestyles, vaccinations for special environmental situations, vaccinations for special health status persons, for travel and against bioterrorism. Okay. Some places require some kind of vaccinations according to before arrival and after departure also. So here we'll talk about a little bit about the COVID-19 vaccines because it's fascinating. So what do the nucleic acids do? We know that it's a human cell, it's a membrane, the DNA is inside the nucleus, which basically gets converted to RNA, RNA gets converted to proteins. The cell DNA is in the nucleus is a code for making RNA. Obviously it's stable, it's the nucleus. And it codes for cell RNA, which is for cell cytoplasm. It's a code for making proteins. It's unstable and falls apart in hours. And it codes for proteins which are found in the cell cytoplasm. Proteins build cell machinery. Now, how do viruses work? DNA virus, protein shell, spike protein, and DNA code. That is how it works. So, virus enters the cell, viral DNA enters nucleus, viral DNA makes viral RNA makes proteins, and proteins make new viruses. Cell burst releasing viruses. The viruses infect new cells. Infection eliminated by the immune response. Okay. Now, what happens for RNA virus? This is the coronavirus. So this has a fatty coat stolen from infected cell, okay, and a protein shell, a spike protein, and RNA code. So the virus will enter the cell, viral RNA will enter cytoplasm, viral RNA will make the viral proteins, proteins will assemble into viruses, cells will release viruses, viruses will infect new cells, and infection will be eliminated by the immune response. So what kind of coronavirus vaccines have been made? So we discuss about Oxford or the AstraZeneca vaccine, the adenovirus vaccine. So basically it has an adenovirus protein shell empty of natural virus DNA. The synthetic DNA coding for coronavirus spike protein is inserted into the otherwise empty adenovirus shell. Okay. Now the virus shell carries coronavirus spike DNA into cell nucleus. Coronavirus spike DNA makes spike RNA and the spike protein cytoplasm. Spike protein is released to activate the immune system. Neither virus shell nor coronavirus spike DNA persist or cause any disease. This is the AstraZeneca vaccine. Now the biotech Pfizer Moderna vaccines, RNA vaccines, have synthetic fatty coat similar to cell membrane. Okay. And it contains synthetic RNA coding for the coronavirus spike protein. The coronavirus spike RNA enters cell cytoplasm. They make the coronavirus spike protein, and spike protein is released to activate the immune system. Neither fatty coat nor the spike RNA cause any disease. Now, how do they stimulate an immune response? So, vaccines make coronavirus spike protein inside cells, which is presented to the immune system as an antigen, right? Now the immune cells will detect the spike protein and make antibodies specific for the spike protein. Now during infection, antibodies bind to spike protein on real virus and protect against infection or disease. So antibodies speed up elimination of the coronavirus from the body. Now, what are some of the conclusions? Oxford vaccine is a protein shell from a harmless adenovirus into which synthetic DNA coding for the coronavirus has been added. So, it's highly purified virus particles without contaminants or additional chemicals, except possibly a harmless preservative. Synthetic DNA from the vaccine persists briefly in the cell nucleus and the cell dies, and the DNA is digested by enzymes. For the RNA vaccines, we made a synthetic RNA coding for the coronavirus spike protein surrounded by a synthetic fatty coat like our cell membranes. The highly purified fatty particles without contaminants containing small amounts of polyethylene glycol. It is also a harmless chemical. Coronavirus RNA persists briefly in the cytoplasm, making the protein and naturally degraded by the enzymes within hours. Now, what are the other considerations? Unlike previous generations of vaccines that are contaminated with other proteins like egg proteins in influenza vaccines or mixed with adjuvants such as alum salts or preservatives containing traces of mercury, these vaccines are clean. There is no known mechanism by which vaccine RNA can interfere with their own genetic code, and there is no evidence from animal studies or clinical trials where they exist that this can occur. So there is no known mechanism by which DNA introduced into the cell by adenovirus can interfere with our own genetic information, and natural adenovirus infections are commonplace and harmless. Okay, so this was the question that we started with. We also learned a lot about vaccines. We also talked a little about the coronavirus vaccine, right? So the correct answer will be protection from infection for which vaccine is made. So anybody has any question, doubt in this question? Okay, so if nobody has any doubt in this question, maybe we can stop the session here today. I will upload the slides and also the Google. I'll upload all these slides and the session recording which I have with me. I will upload that on YouTube and the links will be shared. So we'll meet next Monday, same time, 6 p.m. Thank you, everyone. I'll stop sharing the screen. Thank you, everyone. You can leave the meeting. I will share everything. You can just go through that. Thank you.